Good morning, students. Today we will be discussing about the beta-lactam antibiotics. First of all, we should know what is antibiotic. So, antibiotics is very common drug or common known drug. So, the drug which is effective against the bacteria. So, the bacteria having different structure like spherical, rod shape, bacillus type. So, they have particular cell wall, right? So the bacteria, how the bacteria will be dying in our body whenever the bacteria has incorporated in our body. So the bacterial cell wall or the bacterial genetic material will be destroyed by some antibiotics. So they are called the antibiotics. So one of the category is the beta lactam antibiotic. So beta lactam antibiotic means they have a particular structure, a nitrogen containing, a sulfur containing ring, that is known as the beta lactam ring. See the structure here. It has a 4-membered nitrogen containing ring and a sulfur containing 5-member ring as a thiazole ring, and this ring is effective against some bacteria which will prevent the cell wall. So the beta-lactam ring is a part of the core structure of several antibiotic families like the penicillin, cephalosporin, carbapenem and non-monobactam, which are therefore also called as a beta-lactam antibiotic. Nearly all of these antibiotics work by inhibiting the bacterial cell wall synthesis. So they has a lethal effect on bacteria. That means whenever the cell wall of the bacteria will be destroyed, then the total cell organelle will be destroyed, the replication of DNA or genetic material will be inhibited. As a result, the bacteria will be dying. So the beta lactam antibiotic containing a particular structure. So see, these are the different structure of the bacterial antibiotic. So these structures generally like penicillin, cephalosporin, cefamycins, oxacephams, clavulanic acid, penams, carbapenems, non-cardicin, monobactams. These are different structure, whereas a basic core structure is a 4-member nitrogen containing ring and a sulfur and nitrogen containing 5-membered ring. So coming to the classification of the category, so penicillin, cephalosporin, carbapenem and monobactam, these are the main category, whereas cephalosporin having the 4th generation, 1st generation, 2nd generation, 3rd generation, 4th and 5th generation. So coming to the penicillin, so these are different category, whereas penicillin G, penicillin V, methicillin, oxacillin, cloxacillin, dicloxacillin, amoxicillin, carbenicillin, piperacillin, cefoxitin, these are the important category. So coming to the cephalosporin, these are the primitive category of the drug, whereas first generation like ampicillin, cefadroxil, cephalaxin, cefatolin, cefadrudin. So these drugs having some of the cytotoxic activity and some of the cytotoxic. That's why newer drug have been invented, second generation, that is cefazolin, cefanoic acid, cefametazole, cefuroxime. So these drugs are very specific to some particular gram positive or gram negative bacteria, but there were also some of the adverse effect. That's why third generation, which are newer, where discoveries are going every day, the molecules has been derived with the different derivatives. The third generation antibiotic like cefaclor, cefapirazone, cefitoxime, cefidazidime, ceftriaxone, these are the important category. And now some newer drugs like moxalactam, these are the another category. Next, coming to the newer research, 4th and 5th generation, cefafin, cefozoram, cefiferon, cefiquinone, like these are the important category. Recently, we have been found that different antibiotic resistance are going on. The bacteria are changing their structures day by day. They are hiding their structure or just changing with a sudden, with very short of time. With the antibiotic, their structure is changed. So that's why some of the bacteria is not responding to particular antibiotic. That's why newer generations are going on. So that's why the first generation, second generation, third generation, and fourth and fifth generation innovations are going on. Like next, coming to the carbapenems, that is a biapenam, ertapenem, doripenem, like this. Monobactams, azathionam, triazolamum, and carinomer, no cardicin A. So sometimes it has been found that the different surgery cases or different hospital acquired disease, in some cases of the incubator and intensive critical care unit, whenever the patients are going on the ventilation, some of the bacterial infections are going on. These bacteria having critical structures, they are surviving in a very favorable condition or unfavorable condition also, like they are surviving in so in the super so these bacteria are also surviving in like air conditioned area. So this bacteria's dying, or this bacteria's inhibition or killing is very crucial. That's why this type of higher category drugs are synthesized. Next, coming to beta lactamase antibiotic inhibitors. So penicillin, cephalosporin, this drug containing the beta lactam ring, and acquired resistance by developing beta lactamase. So sometime whenever, so going to the next generation, whenever the antibiotic, beta lactam antibiotic is given to the particular bacteria, they produce an enzyme that is known as the beta lactamase enzyme, which will acquire resistance against the beta lactam antibiotic. So the beta lactamase enzyme, whatever the enzyme is present, which have to be inhibited. That's why some newer drug has been prepared, so they are known as a beta lactamase inhibitor. So let's go about the treat infection caused by the beta lactam resistant bacteria. So like these are the bacteria Haemophilus influenza, which will not be effective against the beta lactam drug antibiotic. Methicillin sensitive Staphylococcus aureus, Bacteroides fragilis, Proteus species, Escherichia coli, Klebsiella species, Acetobacter species. Cephalosporin containing different combinatorial drug, Pseudomonas aeruginosa. This bacteria will inhibit the beta lactam antibiotic. That's why this bacteria will be treated through some beta lactamase inhibitor drug. That means their enzyme will be inhibited by some antibiotics. So as a result, they will not they will be killed, or they will be they will be dying through this drug. Okay. So, beta lactam is a family of enzymes involved in the bacterial resistance to beta lactam antibiotics. They are agents which resemble to beta lactam molecules. As a result, they are making a competitive inhibition with the drug molecule. So, as a result, drug molecule cannot fit into that bacterial structure. As a result, the drug cannot act. That's why some beta lactamase inhibitor drug is synthesized, which will be inhibiting the action of beta lactamase enzyme, like clavulanic acid, sulbactam, tazobactam, avibactam. These are the drugs. Now coming to the beta lactam enzymes, again I'm giving some broad spectrum examples like clavulanic acid, tazobactam, sulbactam. So these are the inhibitor of the beta lactamase enzyme. Trade name, coming to that important trade name, augmentin. That is the amoxicillin, the clavulanate. These are the very important drug available in the market. Josine, piperacillin and tazobactam. Some of the penicillium notatum derivative drugs, and some of the tetanus which is going on. The infection is not treated by some general antibiotics. That's why this drug is given. Timantine, ticaracillin, clavulanate. So coming to the mechanism of action, these three substances resemble beta lactam molecule and are protein inhibitors of most plasmid mediated beta lactamase. That means the beta lactamase enzyme is secreted through the plasmid. So sulbactam has an intrinsic activity against the Acinetobacter and may be used against the MDR strains. MDR strains means multi-drug resistance. That means some of the bacteria having very good potential or resistance to different drugs. So that's why their killing is very crucial. But this sulbactam is very good and in having intrinsic activity. So this Acinetobacter bacteria can be killed. Now indication where the beta lactamase enzyme will be acting. So used in fixed combination with specific penicillin, ampicillin, amoxicillin and ticaracillin. Penicillin beta-lactamase inhibitor combination are used for empirical therapy against a wide range of potential pathogens, including treatment of aerobic and anaerobic infections like intra-abdominal infections. So this type of infection can be cured by these beta-lactamase inhibitors. The beta-lactam inhibitor merely extends the activity of the combined penicillin. That means wherever the penicillin cannot act single way, so then that's why combined drug is given. So, most important are carbonic, sulbactam, and tazobactam. So again, this structure having a very weak antibacterial activity also important in combination with penicillin, sensitive to beta lactamase degradation. That's why these drugs are given with the penicillin, because some of the drug can be degraded by the beta lactamase. So coming to the first, clavulanic acid is the first one of this class, whereas natural product from streptomyces, and it has been derived from this bacteria. This category has a powerful and irreversible inhibition of the beta lactamase enzyme, because it will covalently bind to two positions in active site. And see the structure of the oxygen and nitrogen containing ring, whereas 1 2 3 4 5, whereas 1 and 4 position is the crucial point, that is the active site, and normally used in combination with the amoxicillin, other beta lactamase sensitive penicillin. So coming to the SAR of the beta lactamase inhibitor, see, 1 position oxygen is there, 4 position nitrogen is there, and the 7 position enol group is there, and the 6 position that is free. But in case of beta lactam antibody, in the 6th position there are different derivatives are, which is changing time to time, and the structure of the drug is also changing. Beta lactam ring is essential. The enol either have a role in binding. The alkene moiety should have a Z configuration. See, at the second position beside the oxygen there is a alkene moiety, double bonded carbon, should have a Z configuration, which is much more active than the E isomer. At the C position, at the 6th position there is no substitution, and the R stereochemistry at the 6-3 position there is a COOH group that is below the plane, which should be like this way. Carboxylic acid is at the C3 is very essential, so you have to write this beta lactam inhibitor. So there are some 4 structure of beta lactam antibiotic family. So generally beta lactam inhibitor having the beta lactam antibiotic structure, but in case of beta lactam where the sulfur group is replaced by the oxygen, has negligible antibacterial activity, bind to beta lactamase and protect the antibiotic from the destruction. Most successful when they bind the beta lactamase irreversibly. The important in medicine: clavulanic acid, sulbactam, tazobactam. See the beta lactam structure ring in the green color, whereas sulfur nitrogen containing ring, beta lactam ring, is degraded by the beta lactamase enzyme, as this drug cannot act. So this drug structure has been prevented with the replacement of the oxygen in case of sulfur. So the beta lactam inhibitors, see the clavulanic structure, that is from the streptomyces clavuligens, no acyl amino side chain, sulfur replaced by oxygen. In case of 4th position, oxazolidine ring is present. Oxazolidine because oxygen nitrogen containing 5 member, that's why oxazolidine. Beta lactam weak, unimportant antibacterial activity, powerful irreversible inhibitor beta lactam because suicide substrate, used as a sentry drug for ampicillin, augmentin, amoxicillin, clavulanic acid, allow less amoxicillin per dose and an increased activity spectrum. So these are the important beta lactamase inhibitor drugs: clavulanic acid, sulfonic acid, tazobactam. Many antibiotics like the ampicillin, amoxicillin, tiacrocillin, and penicillin are also available in the combination with one of the several beta lactamase inhibitors like clavulanic acid, sulbactam, and tazobactam. So that means amoxiclavulanic acid. So clavulanic acid is a beta lactamase inhibitor. Okay. So now coming to the beta lactam versus non-beta lactam differentiation. Beta-lactam means antibiotic that have a beta-lactam ring within the molecular structure. Non-beta-lactam means antibiotics are the antibiotics that do not contain a beta-lactam within their molecular structures. They are very powerful, less powerful than the beta-lactam antibiotics. Penicillin derivatives, cephalosporin, monobactam, and carbapenem. These are the example of vancomycin, bacitracin, formycin, chloramphenicol, and daptomycin. So thank you all. Just I think I have cleared about your beta lactam antibiotic and beta lactam is inhibitor drugs. Thank you all.